This is the molecular biology presentation for Azari Mehr, Madelyn and Jade Colley. We decided to base our presentation on sex chromosomes complement regulates expression of mood-related genes, which was published on 7th of November 2013. Major depressive disorder, MDD, is a common chronic illness characterized by lower mood, sadness, irritability, along with physiological changes, ultimately interfering with family relations and normal daily activities. MDD is twice as common in women as in men, with the life incidence of MDD being 20% in women and 12% in men, affecting approximately 14.8 million adults in the United States of America. Interestingly, sexual dimorphism in gamma-aminobutyric acid GABA, serotonin, and dopamine systems are also reported. Understanding the mechanisms behind these sexual dimorphism may help unreveal the biological basis of the heightened female vulnerability to mood disorders. Imaging studies in humans report dopamine-related sexual dimorphism in the frontal cortex of the control subjects, with increased dopamine D2-like receptors in women compared to men. Sex differences in dopamine have been extensively studied in rodents and report, for instance, lower dopamine levels and higher turnover in the frontal cortex of female compared to male rats.
A three-way analysis of variance was used to determine if developmental gondal sex and circulatory hormones had any correlation between gene expression and anxiety-like behaviour. The results were compared to mice with ovaries and mice with testes. All of the mice were given a gonadectomy at 10 weeks prior to being euthanized. Any gene expression differences would have been due to the hormone exposure before the experimental period. A number of tests were carried out, 13 for GABA-related genes and 14 for serotonin dopamine-related genes. The contrast of these two variants was determined by the Turkey's post-hoc test. This determines which groups in the sample differ. This test is different from the ANOVA test because it is carried out after the analysis of variance. The results of the post-hoc test showed that mice with ovaries during development and that had been treated with testosterone in adulthood had higher cortistatin levels than blank-treated mice. Heat maps were created to show the correlation between gene expression and anxiety-like behaviour. The male phenotype was divided by the female phenotype. The heat map showed that if the expression of a gene was higher in the male phenotype, the heat map codes for the colour red. And if the expression of the gene was higher in the female phenotype, the heat map codes as blue. When looking at the effects of sex-related factors on expression of serotonin and dopamine-related genes, it showed that circulating testosterone caused a decrease in 5-hydroxytryptamine expression in gonadal males, but not in gonadal females. However, 5-hydroxytryptamine expression was lower in gonadal males when they were treated with testosterone. This showed a significant interaction of circulating testosterone and gonadal sex. This circulating testosterone also increased dopamine expression in gonadal females, but decreased in gonadal males. The correlation between gene expression and anxiety-like behaviour showed that anxiety-like behaviour did vary due to sex-related factors. XY mice exhibited reduced expression of genes coding for markers of dendritic targeting GABA interneurons and for GABA synthesizing enzymes. XY mice also displayed a higher state of anxiety-like behaviours compared to XX mice. These findings partly mimic changes observed in human subjects affected with major depressive disorder, which supports the theory of a contributing role of reduced dendritic targeting interneuron-mediated inhibition to the respective behavioural phenotypes. Findings of reduced GABA, serotonin and dopamine-related gene expression in XY mice show that sex chromosome complement causes expression to be lower in individuals with male sex chromosome. Women are more vulnerable to mood disorders, which suggests that additional factors in normal males may compensate for this, for example, protein expression or other independent factors. Even though the results did not clearly identify this specific factor, there was non-significant compensation by a number of genes due to male gonadal sex and testosterone. Future studies should aim to identify genes and proteins where expression is altered in anti-disease directions by circulating testosterone treatment. Previous studies have provided further evidence for the role of GABA, serotonin and dopamine in psychiatric and neurodegenerative disorders. These studies provide strong evidence that the decreased expression of genes related to GABA, serotonin and dopamine signaling is related to the XY sex chromosome complement. Combined with previous results indicating heightened anxious behaviours in XY mice and evidence for the involvement of this in the pathology of mood disorders, these results provide molecular leads to investigate the biological basis of gender-related differences observed in mood disorders and to specifically identify male XY sex chromosome complement as pro-disease factor. Therefore, these findings have broader implications for the understanding of gender differences in adults